Hi, my name's John, and my partner's name is Josh. Our lab was on the effects of increased heat shock time on transformation through Mercyhurst University. Genetic transformation. This is the adding of foreign DNA from a different species into a cell. The cell we transformed was calcium-competent E. coli with pea-green plasmids. The calcium ion in the solution neutralizes the negative charge of the DNA backbone and the negatively charged phosphate in the plasmid membrane. This allows for the DNA to interact with the surface of the cell. This makes the cell able to take up the plasmid upon stimulation of heat shock. In the picture to the right, it's going to show this genetic transformation of the foreign DNA on the left, labeled BDDAC, entering into the cell and then joining up with the DNA of the original host cell. Our plasmid was the pea-green plasmid. It has an addition of a selectable marker, AMP-R, also the addition of a multi-cloning site of GFP, green fluorescent protein mutation. The AMP-R ampicillin resistant encodes enzyme beta-lactamase, allowing it to break the beta-lactam ring, making it resistant to beta-lactam antibiotics like ampicillin and penicillin. And the GFP mutation, which is derived from a bioluminescent jellyfish, Aquaria victoria, which allows the bacteria to glow a greenish yellow under UV. And in the picture behind, you can see an example of this GFP mutation and the glowing color it represents and gives off. Our variable for this lab was the heat shock time of 3 minutes. During the heat shock stage, it opens micropores in the membrane, allowing for the uptake of plasma DNA. The original heat shock time was 90 seconds, and for the experiment, we had doubled the time. With the overexposure of heat, we feel as though it could lead to the possible damage of the host cell, risking its chances to take up the DNA. Too long of heat exposure could also cause the overexposure of DNA and prevent the uptake of it. Higher temperatures of heat shock have still shown the capability of transformation of DNA, but no studies were able to be found with the time of heat shock increase. Our hypothesis is the extended heat shock may overexpose the cell and do the reverse effect to allow DNA into the cell.
For the results section, the table on the right corresponds with the picture on the left, showing the colony growth on the plus plasmid plates and no growth on the variable plates due to an absence of pea-green plasmid in the variable samples, and so they were not able to carry out transformation. So the transformation efficiency for the plus plasmid we found was 2.4 million colonies per microgram per milliliter, and we were not able to calculate the full difference due to not knowing the transformation efficiency of the variable, as there was no transformation. In conclusion, if the experiment was carried out correctly, it would be expected that the transformation would have been less than the plus plasmid. And we found that the heat shocking for a longer period equaled less transformation of the cells. And this could have been because of the viability of the cells decreases with longer exposure at high temperatures. We found in the Singh et al. Paper. And the original hypothesis that was formulated was expected. The overexposure to the heat shock could decrease the viability of the cells and reduce the transformation efficiency. So we agree with our hypothesis that we chose. And these are the references that we used for our presentation.